Good afternoon, everybody, and welcome to this ESID Grand Round webinar on congenital thymic aplasia and thymus transplantation. We have a fairly packed program this afternoon. We're going to be talking about treatment of thymic disorders and the diagnosis of thymic disorders. And as you can see, we have a distinguished list of speakers and panelists. And in the interests of time, so that there's plenty of time for questions, I am going to hand over to Fatima, Dhalla, sorry, to Alexandra Kreins, I think is starting first, to kick off the session. So welcome, enjoy. If you have questions, please put them in the Q&A, and hopefully there'll be plenty of time at the end to do that.
Okay. Thank you, Andy, for the introduction to this grant round. So we thought it would be interesting to start with a poll to have an idea of the experience or the clinical experience in the management and the treatment of patients with congenital athymia. And so if it were possible, so here you can see the first poll, and so you have an option to let us know whether you have, in your experience, already referred a patient for thymus transplantation, or if you've been involved in treating an athymic patient, but with hematopoietic stem cell transplantation, or if you've looked after patients who've been treated with thymus transplantation or hematopoietic stem cell transplantation, or if you have picked up an athymic patient via newborn screening, or option E, if so far you have no experience in diagnosing or treating any patients with congenital athymia. Okay, thank you. So it's quite interesting to see that for about 60 or more than 60% of the audience, there's actually no experience really in the management of patients with congenital athymia. And so we hope that the session we've prepared will be very informative. And so to start with this session, we will have a first clinical case. So Graham, if you're happy sharing your slides. Okay, so we thought we'd start with a clinical case. And so this is a patient who was referred to Great Ormond Street Hospital, which has a thymus transplantation program. And the patient was referred as a possible candidate for thymus transplantation. This patient was picked up by newborn screening shortly after implementation of the universal newborn screening program for SCID in Germany. And so this patient didn't have any TREX, and this was confirmed by immunophenotyping, which showed profound T-cell lymphopenia, but normal B-cell and NK cell counts. Clinically, the patient did have the Georg syndrome features, including a mild heart defect, but the patient was hemodynamically completely stable. The patient was found to have hypocalcemia and was diagnosed with primary hypoparathyroidism, and also had some discrete facial dysmorphic features with retrognathia and low-set ears. From a genetic point of view, so through FISH, no deletion was detected on chromosome 22q11, but a clinical exome found a heterozygous mutation in CHD7, and so the patient was diagnosed with congenital athymia in the context of complete Georg syndrome due to charge. And so at this point, we have a second poll. Thank you. So, which is gives you an opportunity to let us know how you think this patient should be managed, with several options. So the first option being whether we should proceed with allogeneic hematopoietic stem cell transplantation within 3 months, as per newborn screening skip guidelines, or if the patient should be treated by allogeneic thymus transplantation, but only if no matched sibling donor or matched family donor has been identified, or if thymus transplantation should be considered as a first-line treatment, or if you would propose to just proceed with supportive management. Okay. So see that the majority, so 84% have voted for thymus transplantation as first-line treatment. And so in the fact, we did obviously accept the referral of this patient for thymus transplantation at Great Ormond Street Hospital. And so I leave the word now to Graham Davies, who is the director of the thymus transplantation program at GOSH, and who will tell you more about our program here.
Right. Hello, everybody. So thymus transplantation has been around for quite some time. It was first developed at Duke University by Louise Marcotte. And then we did our first patient in 2009 in London. I think there's an important point of distinction between these two centres that are the only two centres at the moment performing this treatment, and that is that thymus tissue for transplantation in the USA is considered a drug and is licensed by the FDA, whereas in Europe it's considered a tissue for transplantation and regulated in our country by the Human Tissue Authority. So since 2009, we have done 61 transplants in 59 patients. So 2 patients had two transplantations. And on the left, you can see the breakdown of the diagnoses of the DeGeorge phenotype. And there's an important point here that we call these patients complete DeGeorge syndrome, whatever, if they have the phenotype, whatever the molecular cause. So it isn't restricted to just 22q. It includes charge TBX1 and so on. We've also treated some non-DeGeorge phenotype cases of athymia. And on the right, you can see the breakdown over the last few years, showing that in the last few years, despite the pandemic, the number of patients that we've treated has gone up, and I'll discuss some of the possible reasons for that. What I have analysed here in this slide is the age of transplantation for the first 10 years of the program, and then for the last 3.5 years. And you can see that the median age of transplantation has come down to 5 months. And that might be, at least in part, to the introduction of newborn screening. And we've had 9 patients referred who were picked up on newborn screening. Interestingly, 5 of them came from Germany, who introduced newborn screening, I think, in September 2019. We looked at the number of infections. Often in the past, these patients have had infections on board when they came to us, and that has dropped a little, perhaps disappointingly, not as much as we would have hoped with the earlier diagnosis and referral. And interestingly, despite newborn screening, 2 cases managed to acquire CMV, presumably because of breastfeeding in that window whilst waiting the results of the newborn screening. For this analysis, I've excluded 3 patients that we transplanted, 2 who had had prior BMT, and 1 who's going to be presented later, there was a late treatment for FOXN1 deficiency. So what about the outcomes? Well, I think that this slide illustrates that, that those patients who do well end up with a good quality of life. They are able to come off prophylactic medications and immunoglobulin, attend school, and lead a reasonably satisfactory life. Of course, they do have the comorbidities that go with Georg syndrome or whatever condition they have. Their immune reconstitution is slow. This is a typical case showing the recovery of the different T cell fractions, and you can see that nothing much happens for the first 6 months, and then things take off later. We are very keen on doing, for reasons which I'll explain a little bit more later, we're very keen on doing biopsies at 2 or 3 months, because at that stage, even though there's nothing in the blood, one can see evidence of thymopoiesis developing with cortical thymocytes expressing CD1A, the cytokeratin pattern, and here is the thymus in the muscle, under the capsule of the muscle, looking just like a normal thymus. So reconstitution is slow. The median time in eligible patients to reach what we consider a minimal naive CD4 count of 50 is 9 months, but some take much longer, some are quicker. On the whole, the very slow cases reconstitute slowly because they've got some other comorbidities, such as a viral infection or inflammatory or autoimmune disease, and this may be due to the underlying condition or to the immunosuppression that they may need. For those that when recovery occurs, they usually plateau out at about 18 to 36 months after the transplant, and these are in those patients who've achieved that timing, these are the levels of the CD4, naive CD4 and TREX that we see, and with the lower limit of normal given underneath. And you can see that although there is reconstitution, it is not usually to the normal range for the age of the child. If we, by chance, some of the patients, these transplants are not HLA matched, but by chance there is sometimes some matching. And in this slide, I've shown the effect of the maximum TREC level achieved after transplantation of having 2 class 2 alleles matched, so 2 of the 4 DRTQ alleles matched, and you can see that there's a trend for the TREX to be higher if there is 2 high resolution matches, that's 4-digit matches. But even with allelic matches, there's a bit of a trend, and certainly there are some children right down the bottom there that in the ones without any matching. So we think that possibly, although this is not statistically significant, that possibly may indicate that would help, and that makes intuitive sense. What about complications? So as I said, many children sail through, but some do get problems. An inflammatory disease is something that we don't really understand. It occurs typically at the time of immune reconstitution, so about 4, 5, 6 months out when things are starting to happen. And in its mildest form, there's a mild rash, which usually settles spontaneously. But in 6 of our patients, we saw quite significant inflammatory disease. In some of those cases, this was driven by infection, and it was focused on the organ that was infected. So protracted enteropathy when there was some viral infection in the gut, and a bad skin infection in the child who developed HHV6. But in 2 patients, we saw no identifiable driver, and they got a very severe systemic immune response with quite bad consequences on the outcome. Autoimmunity is also an issue. 35 patients have reached more than 18 months, and we've looked at them. 21 of that 35 had at some stage definite autoimmunity. With this inflammatory disease, it's sometimes difficult to be absolutely sure what's autoimmune and what's an inflammatory dysregulation. 4 of those cases were very transient and early before immune reconstitution and have not recurred, and that might be considered similar to what happens in a bone marrow transplant situation, where early on you get dysregulatory cytopenias on occasion. And then later disease was predominantly affecting the thyroid gland, both hyper and hypothyroidism, and all cytopenias, with occasional patients who've had renal or neurological autoimmunity. One patient actually had cytopenias and neurological autoimmune problems before transplantation, and I think that may be a clue that Georg patients are prone to autoimmune disease, and it may be because we're not fully reconstituting them, we're converting them from complete Georg to partial Georg, and they retain their susceptibility to autoimmunity. We've also looked at the effect of chance class 2 MHC matching, and there is, again, a trend that those patients with autoimmunity were more likely to have no class 2 matches, whereas those who didn't have autoimmunity were more likely to have 2 loci matched. And you can see one locus has no effect in that regard. It's just 2 loci. The mortality in our group was just under 20%, so 11 patients died. Sorry about the typo there. 10 died before they got immune reconstitution, and 1 child died of late immune thrombocytopenia with cerebral hemorrhage. The early deaths were often associated with pre-existing viral infections or viral infections acquired while waiting for thymopoiesis to develop, and often these drove inflammatory disease, and there were a number of other causes of death. In addition to the Georg patients, we transplanted 3 patients with FOXN1 deficiency. The first one is going to be presented just after I finish speaking, so I won't say anything more about him. We transplanted a girl with 2 null compound heterozygous mutations early, and she was very unlucky in that she had a positive biopsy at 4 months, and before thymopoiesis really blossomed, she developed HHV6, terrible exfoliated skin rash associated with the dysregulation that went with that, and she died after immunosuppression of aspergillus. And then one little girl at the bottom who, despite having CMV, pneumonitis, and CMV in the central nervous system, did well and is thriving with good reconstitution. 2 patients with PAX1 deficiency have been transplanted. The first was post-bone marrow transplant and was in a very poor state, having had a 4 out of 6 cord blood transplant, resulting in severe graft-versus-host disease with cytopenias and to capit norovirus and gastrointestinal failure. And she didn't do well. And she illustrated the problem that certain comorbidities will cause. We knew she had some vertebral abnormalities, but they weren't causing any problems. But 6 weeks after the transplant, the spinal cord became compressed because something shifted. and she had to have emergency neurosurgery, which obviously wasn't the ideal time for that to happen. She died of sepsis whilst on immunosuppression. And then one more recent patient diagnosed on newborn screening has responded to treatment and done well, although he does have some thrombocytopenic episodes. We have done two cases who previously had hematopoietic stem cell transplants because they were thought to have a bone marrow type of severe combined immunodeficiency. Louise Market has reported 4 such cases from her series in the United States, and she's pointed out the importance of matching of the donor to any host HLA antigens mismatched to the stem cell donor for fear of exacerbating graft versus host disease. So the 2 that we've done, one was the PAX1 patient that I just told you about that was unsuccessful, and then we treated a non-DGS undefined athymia, so no DGS features, who'd had a matched sibling transplant, so matching with the donor and recipient was not so important. She's had very slow immune reconstitution, but seems to be getting there. So this does work, but it's not to be recommended. And so early diagnosis of whether the cause of absent TREX on newborn screening is due to a bone marrow disorder or a thymic disorder is very important, so that the right direction of treatment can be chosen. And to that end, tests looking at the use of OP9-DL1 assays or similar notch ligand assays to drive T cell development can, in most cases, we think, distinguish thymic from non-thymic T cell deficiency. And we're grateful that we've been collaborating with Isabelle André in Paris to do that, and now to set it up in our laboratory. I'd like to quickly finish by acknowledging my colleagues and the team, these first 2, Alex and Evie. Anybody who's interacted with us on the Thymus team, that they do the lion's share of the work. Other members of the staff, including the surgeons who put the Thymus in, and our collaborators. Big thanks. The list has now got so large I can't present it, but we have co-managing clinicians who do an awful lot of the work looking after these patients after transplantation when they get complications, mainly across Europe, but as far away as the Antipodes, and of course the patients and families. And Evie is our clinical nurse specialist who has worked very closely with the patients and families to write with them, really, with a lot of their input, some new guidance booklets outlining what the treatment involves, a very simple one and a more complicated one, which is what the families wanted. And those will be becoming available soon. Those are some grant research money that we get to support this work. And I'll finish by going back to that first case that Alex presented. So this patient was referred for thymus transplantation, as I think the poll largely suggested most people would do. The thymus transplantation took place at 11 weeks of age. There were no atypical features, by which there were no Omenzi features, so no ATG was needed, and it all went very smoothly. And he went back to Freiburg after 2 weeks or less, 21 months out with pretty good immune reconstitution, and this was his biopsy showing good thymus with CD3 and, in this case, Ki67, another marker of proliferating thymocytes. Okay, I'll stop there and look forward to having some interaction back. Thank you very much.
Graham, will you stop sharing your screen? Yeah, great. Okay. So my name's Fatima. I'm a clinical immunologist. I work in Oxford, and I also work in Georg Hollander's lab, where we study kind of basic aspects of thymic stromal cell biology, and we collaborate with the folks at Great Ormond Street with regards to the kind of research aspects of cases with thymic stromal cell defects. So for the next part of the session, we're going to present some more clinical cases, then hand over to Georg Hollander, and Alex and I will conclude the clinical cases at the end. So the next case is relates to a girl who was referred to Great Ormond Street when she was 2 months of age with a kind of omens-like phenotype. So she had severe dermatitis, diarrhea, she had a respiratory tract infection with rhinovirus, and she was found to have severe T-cell lymphopenia. She had basically no naive T-cells and absent TREX. She had a TCR spectrotyping result that was consistent with Omen syndrome, so with oligoclonal expansions and a restricted repertoire. And an ultrasound scan of her chest had revealed that there was no thymic tissue. She did have normal hair and nails, and we ended up finding a heterozygous mutation in FOXN1 in this patient. So this mutation results in a deletion of a single base pair in the C-terminal domain, which is responsible for transcriptional activation. And essentially what happens is there's a frame shift, so you get a non-canonical kind of scrambled sequence between amino acid position 457 and an early truncation at position 550 of the protein. So because of this problem in the transcriptional activation domain, we hypothesized that this variant would be transcriptionally inactive. So with that information, we have a next poll regarding how people think this patient in particular should be managed. So should she have a stem cell transplantation? Should she have a thymus transplant as a first-line treatment is option C, or only if there's not a matched sibling or family donor? Should she have supportive management, or do we not know at this stage? Okay. So I guess in summary, most people think that she should have a thymus transplantation at this stage, and the second most common answer was we don't know yet. So we'll present the conclusion of that case later on. So a third case, this was a patient that was referred to Great Ormond Street when he was 10 years old for consideration for thymus transplantation. Actually, he had a longer history. So he was born with alopecia totalis and nail dystrophy. He had quite significant viral infections, including chronic EBV, which led to an EBV-driven lymphoma at the age of 6. He had autoimmune cytopenias, interstitial lung disease. He also had a T-cell lymphopenia with absent naive cells, absent recent thymic emigrants and TREX. And in this patient, we found a compound heterozygous mutation in FOXN1. So he inherited both alleles from each of his parents. There was one, the P82X variant, that basically leads to a very early truncation, and we predict that to have a null effect. It may not even be expressed at the protein level. And the second mutation, P350L, is a single amino acid substitution in the fork head domain. This domain is important for DNA binding, and we thought that that would probably have a hypomorphic effect. So it'd be interesting to know if people think that the management for this case should be different in comparison to the first case. So we have another poll with similar questions. So whether the patient should have a hematopoietic stem cell transplantation, a thymus transplant, supportive management, or we also don't know. Okay. Okay, so that's interesting. So a similar amount of people, slightly less, think that this patient should have a thymus transplantation, but more people think this particular patient should have a hematopoietic stem cell transplant. Okay, thank you. And we have another clinical case that Alex will present.
Thanks, Fatima. So yes, this is a PAX1 deficient patient who was referred to us from a team in Istanbul, so in Turkey, again in consideration for thymus transplantation. So this patient was born to parents with high consanguinity in the family, and he had an older sibling who had died around 15 months of age, already with a suspicion of some, at that stage, an undefined immunodeficiency. So when the patient was referred to us, they had made a recent diagnosis of PAX1 deficiency, but this patient so clinically presented with some syndromic features which fit with autofacial cervical syndrome, so with PAX1 deficiency, and additionally also was diagnosed with hypoparathyroidism and developmental delay. Initially, the patient presented at 6 months of age with lymphadenitis. No biopsy was done of the lymph node, but it was suspected BCGitis in the context of a history of BCG infection. And the patient then had multiple hospital admissions with another significant event when he was 12 months old, where he was admitted with a lower respiratory tract infection, but the pathogen remained unknown. And then a couple of months later, again with pneumonia and then generalized lymphadenopathies. And at that point, viral PCRs were positive for EBV, but also for CMV. So this patient so far was managed or has been managed from in a supportive way with immunoglobulin replacement therapy, with antimicrobial prophylaxis, including anti-BCG prophylaxis, and then had a treatment course for his CMV, which then ended up not being detected after a couple of weeks, so that he was switched to prophylaxis with valganciclovir. So with the diagnosis of athymia, his team then referred him to our center to consider thymus transplantation, and from an immunophenotyping point of view. So he certainly fits the criteria with absent thymic output, as you can see, with absent naive T cells and negligible TREC levels, normal spectrotyping. However, when we repeated the viral PCRs, while CMV still is not detectable, you can see that there's a very high level for EBV. And so the next poll question is about what the audience thinks in terms of management of this patient. So whether the patient should be considered for a BMT with the best available match, or a BMT only if there is a matched sibling donor or a matched family donor available. Whether we should proceed with accepting this patient for thymus transplantation, or option D is to continue with the supportive management, or option E, whether to treat first with rituximab. Okay. Okay, so you can see that about 44% of the audience is suggesting to proceed with thymus transplantation, but then also similarly, to first treat with rituximab, or a little bit a lower proportion to suggesting to proceed with BMT with the best available match. And again, we'll get back to this case after the presentations that will follow. Thank you.
So, good afternoon, everyone. I'm Georg Hollander. I have the pleasure to provide you with some of the immunobiology background of the thymus, now that most of you have chosen the thymus as a possible tissue to be transplanted. As you know, the thymus is the primary site of T-cell lymphopoiesis. And if we do a cross-section through the thymus and you look at the schematic representation, then you can see three anatomically different areas: the cortex, the corticomedullary junction, and the medulla. And the big vessels are in the corticomedullary junction, where the homing of hematopoietic precursor cells occur once the thymus has been vascularized. From there, the precursors move into a microenvironment where their commitment to T-cell fate occurs. They start to expand, everything still in the cortex, and then start the journey through the depth of the cortex all the way back to the corticomedullary junction. And it is during that time that they are positively selected as well as negatively selected, whereby negative selection takes care of potentially autoreactive T cells, and this is a mechanism that is in place both in the cortex as well as in the medulla. After negative selection has been completed in the medulla, the T cells will have to further mature to a functional competence that corresponds to the one that they display in the periphery, and only once that post-selectional maturation has occurred, then the T cells are able to exit to the periphery. And this is the process that, as you all know, occurs throughout life. And even in octogenarians or older individuals, we still have some thymus tissue that produces new naive T cells. However, what is very important is that the thymus it provides an environment where the T cells differentiate in a non-autonomous fashion, whereby characteristic tissue architecture allows for this complex progression from an uncommitted T cell to a mature T cell, and where the stroma is largely composed of thymic epithelial cells, but not exclusively. And so if we look at a immunohistological staining of a mouse thymus, then you can see in the outer part a gray zone, which is cortical thymic epithelial cells, which are stained here with a pseudo color in gray. We have the lively medulla in the middle of the picture, where different cytokaratins identify different populations of thymic epithelial cells. And then we have, in addition, just to show you a little bit more about the complexity, fibroblasts, endothelial cells are the mesenchymal cells that form as a composite the thymic microenvironment that constitutes the only tissue in the body that is competent to dictate and control the correct T cell development. If we look at the stromal compartment in a little bit of a closer fashion, and here is the use of model systems such as mice extremely informative, then we can differentiate these thymic epithelial cells, or TECs, into cortical thymic epithelial cells, immature, mature, and post-mature or geriatric medullary epithelial cells. And when we then look over time in a mouse life, then you can see that there are some changes over time in the relative representation of the cortical versus the medullary epithelial cells, with over time the cortical epithelial cells being the most dominant subset of, or subpopulation of epithelial cells in this analysis when flow cytometry is used. However, as we all know, flow cytometry is only one way to look at the complexity of individual cell types. And if we analyze the individual thymic epithelial cells using now single-cell transcriptomic analyses to characterize the transcriptome of individual cells, then you will see that the picture is actually much more complicated. So we can easily identify 9 different subtypes of thymic epithelial cells, shown here in the different colors. And when we link them to a cell-sorted population, as defined by the typical FACS-based identification of thymic epithelial cell, then you can see on top that the green population is typically found in FACS-defined thymic epithelial cells, while, for example, the green, dark green population is found both in cortex, immature, mature, and in part the geriatric MTECs. So basically telling us that the FACS analysis is good, but not good enough, and that we are dealing here with actually a much more complex thymic epithelial cell makeup. Based on the transcriptome of these individual cells, we can now define them in a semi-functional way, with on top of this zeta-shaped curve, the perinatal CTECs, which are a phenotype that is present very early in development, but then slowly disappears, and other phenotypes that belong either more into the cortex or into the medulla. And most importantly, even with this analysis, we clearly see that over time, with age, you see a change in the relative frequency of these cells. Now, the description of these different thymic epithelial cells will need to be understood and appreciated in the context of development. And if you look at the development, in this case, of a mouse, then thymic epithelial cell development starts around day 10 of the mouse, which has a gestation of 20 days, or in the humans, it's probably closer to gestational week 8. If we cut a section across the neck and look in the foregut and have then a section that is 90 degrees to the section shown here, then what we can see in a mouse embryo at around day 10.5 is the foregut, which is this green tube that is formed. And from these foregut in the orange circle, you can see now a protrusion, which is the pharyngeal pouch, and namely the third pharyngeal pouch. And that is where the endodermal lining forms the cells that eventually detach from the continuum of the foregut, formed by 11.0 a thymus Anlage, which subsequently differentiates further, and by E12.5 you can appreciate that a thymus Anlage has been formed. Now, most importantly, for this development to occur, FOXN1 is absolutely required. And FOXN1 is the master regulator of thymic epithelial cell development. And without FOXN1, as you know, things don't work. Now, FOXN1 itself as a transcription factor has a N-terminal end, which probably wrongly is defined as the Tech differentiation end. It has in the middle a fork-head domain, which functions as the DNA binding part of the FOXN1 molecule, and then it has a transcriptional activation domain. Interestingly, the fork-head domain recognizes a relatively short motif of DNA, and it is based on 6 nucleotides that are recognized. But when we did the crystal structure of the FOXN1, then you can see here with the red parts of the molecule linked with green parts that the alpha domains of the fork-head structure fit very snugly and nicely into the larger groove of the DNA double helix. And here the interaction allows for the transcription factor to be then transcriptionally active. Now, the FOXN1 molecule as such is somewhat a very interesting transcription factor, because if you look in immunohistology as shown here, be it in the upper row in the cortex or in the lower row in the medulla, you can see that the FOXN1 shown here in a red color forms actually these speckles. And if we look at a little bit closer at the nuclei with regards to these speckles, we can identify that multiple of these speckles are actually formed. To make sure that this is not an artifact of the immunohistology, Joanna Rota in my lab labeled wild-type FOXN1 with a GFP tag and expressed it in a thymic epithelial cell line 4D6. And what you can see here is the nucleus. And again, we find these speckles. So we were particularly interested in finding out what are these speckles and how do they come about and have they any functional role. And to do that, Joanna took fluorescence recovery after photobleaching approach, FRAP approach, whereby you can look at a single cell very close up, and in the center of the objective shown here is the nucleus. And within the nucleus, you can actually see fluorescence moving. And when we did that, what you can see here in the movie on the right, with the circle and the arrow pointing it, we can see these particular speckles forming. And if we shine a very bright light, then the green fluorescence is bleached. After a while, the intensity of the green fluorescence comes back. And this is a very dynamic process because the bleaching of the fluorescence is irreversible. And so with some computational trickery, we can then show how long does it take once photobleaching has occurred for the GFP intensity to return. And this is a dynamic function because the GFP intensity can only return if new molecules that are not bleached from around that aggregate are incorporated into the aggregate, telling us that these aggregates are actually very dynamic structures. Now, why do I tell you that? These aggregates are dynamic structures, and this dynamism really depends on the full length of the FOXN1 C-terminal tail of the molecule. You see here 3 different mutations. Don't worry about what these mutations are. And second to last is the wild type. And when you look at the condensate recovery rate, so how long does it take after I bleached such a condensate for the condensate to return to its original intensity, you can see if I start to modify the thymic, the FOXN1 C-terminal tail, this recovery rate is defective. Now, FOXN1 is a molecule that doesn't work on its own. It possibly associates with other molecules, and to test that and prove that, what Joanna did is she immunoprecipitated wild-type FOXN1, and what she brought down was analyzed by MSMS and identified then various binding partners to FOXN1. These binding partners are responsible for DNA binding or RNA binding, also importantly are involved in RNA splicing, or are regulating transcription by RNA polymerase. And we identified a number of these molecules also functionally. I just show you the data here. One is a molecule called YBX, the other one is a molecule called CBP. And when we use an experimental system where we have cells that normally express a fluorochrome, here YFP, under the promoter beta-5T on the left, that is sensitive and controlled by the expression of FOXN1, then we have a system whereby the CMV promoter constitutively expresses FOXN1. FOXN1 binds to the beta-5T promoter and drives YFP. And now on that background, we can then use CRISPR-Cas9 to knock out particular target genes. And if we do that, you can now see that if we only use vector 1, the vector alone, there is no inhibition, which is on the x-axis, of YFP expression. If we knock out FOXN1, then the system obviously doesn't work. But if we knock out CBP or YBX1 to various degrees, somewhere between 35 and 70%, the expression of YFP is reduced, hence identifying additional proteins that need to be in play to interact with FOXN1. And data not shown here, but important to remember, this interaction with YBX1 and CBP occurs in the C-terminal domain, which we had shown previously and which I had demonstrated is so important for this condensate formation in the nuclei. So I've given you some molecular background and some clues to hopefully understand some of the patients that were presented earlier. And I give back to Fatima.
Okay. So to finish off, I was going to talk a little bit about known and novel inborn errors of thymic stromal cells, and then Alex and I will conclude the clinical cases, bringing in a bit of the information that Georg has given for the FOXN1 defects. So inborn errors of thymic stromal cells can result from defects affecting any part of this developmental and functional trajectory of thymic stromal cells. So if we take the first section, which relates to defects that may cause abnormal third pharyngeal pouch patterning in initial stages of thymic organogenesis. Clearly, the most well-known is deletions at 22q11. This is the most common cause of DeGeorge syndrome, which describes the triad of congenital heart disease, congenital hyperparathyroidism, and thymic hypoplasia or aplasia. Within the deleted region that is deleted in 22q11, sits TBX1, which is a transcription factor. So defects in TBX1, single gene defects, can also cause impaired thymic organogenesis. There are several other chromosomal syndromes and several other single gene defects that can cause abnormal third pharyngeal pouch patterning and initial thymic organogenesis. And added to this, there are a number of environmental factors such as under or overexposure of the fetus to retinoic acid, maternal diabetes and alcohol consumption that can also impair this part of early thymic organogenesis. Actually, many of these factors, the single gene factors, are transcriptional regulators, and they all functionally interact in the process of pharyngeal pouch patterning. Next, when we think about defects that might affect particular stromal cell development and function, clearly FOXN1 is our example here. And further down the line, when air is deficient in a subset of mature medullary thymic epithelial cells, negative selection of overtly autoreactive developing thymocytes is impaired, and this leads to a failure of central tolerance induction and autoimmune polyglandular syndrome. And then finally, novel defects can affect theoretically any part of this pathway. So I'm now just going to concentrate a little bit on telling you about novel defects, because I think this is quite relevant, and a little bit about the defects that we've presented on today. So when we think about novel thymic defects, they fall into two categories by my mind. So firstly, you could have novel variants in known genes, or you could have variants in novel genes, or actually a genetically undefined congenital T-cell lymphopenia. Fortunately, we have at our disposal a number of tools that can help us dissect such cases. So firstly, if you have a candidate variant, you can use in silico modeling to model the effect of a genetic variant on protein structure or function. And this can encompass simple to use online tools or more sophisticated methods that are either based on solved or predicted 3D protein structures and known structure function relationships. And basically all of these in silico tools draw on things like evolutionary conservation of amino acids, their particular biophysical properties, if we know anything about their relevance in protein-protein interactions, and how mutations affect stability or folding of proteins. And these are the sort of things that come into play with in silico modeling. Now, when we think about novel variants in known genes, well, one would assume, and certainly this is the case for FOXN1, that we know more about structure-function relationships to assist in silico modeling. And because a lot of the single gene factors that have been associated with thymic defects are transcriptional regulators, transcription factors, we have a number of reporter assays at our disposal where we can test in vitro the functional effects of variants. Then moving on to the second category, so the more novel genes or undefined conditions. Graham has touched upon this, but I think it's worth just spending a little bit longer, because such patients are increasingly identified because of our increasing use of whole exome and genome sequencing, and because of the adoption of newborn screening in many countries. So genetically undefined T-negative SCIDs are being picked up, and these patients represent a real therapeutic dilemma, because the options here where T cells alone are defective in their development is whether the underlying cause is a hematopoietic cell intrinsic defect or a thymic stromal cell defect. These patients often need definitive management, and the options here for best management are distinct. So hematopoietic stem cell transplant versus thymus transplantation. Fortunately, we do have now at our disposal a number of in vitro differentiation assays that can help decipher whether a defect might have its origins in the hematopoietic cells or thymic stromal cells. So as Graham mentioned, you can first ask the question as to whether patient-derived hematopoietic stem cells are intrinsically able to differentiate into T cells. So this is asking the question about whether there is a hematopoietic cell intrinsic defect. And there are various co-culture systems that you can use where you would put patient hematopoietic stem cells in culture with stromal cells that express notch ligands that are required to commit these cells to the T cell lineage. And you can either do this in 2D systems or actually in the context of 3D artificial thymic organoids. And so you culture these cells for several weeks, and then you take the hematopoietic cells and you analyze them and see how they've differentiated via flow cytometry. So I just wanted to show some examples. So if we take hematopoietic stem cells from a patient with X-linked SCID. So this patient has a hematopoietic cell intrinsic defect. Their cells are unable to intrinsically differentiate into T cells that express CD5, CD7, TCR, the TCR or CD3. Therefore, in the context of these sorts of assays, if you get impaired differentiation of patient cells, the implication is this is a hematopoietic cell intrinsic defect, and the patient is best treated with a hematopoietic stem cell transplant. In contrast, if you took hematopoietic stem cells from a patient with DeGeorge syndrome, these cells aren't intrinsically impaired in their ability to differentiate into T cells. They just lack the normal endogenous microenvironment of the thymus. So actually, when you look in vitro, DeGeorge syndrome patient cells are able to differentiate into T cells. Thus, by and large, if this sort of assay gives you a normal differentiation readout, you may think whether this patient has a thymic stromal cell defect. Now, there are several important caveats to this. So it's been shown that cells from patients with ADA SCID can actually differentiate normally in this assay. And presumably, if you had a patient with a very late differentiation defect or a more functional defect, they may have more normal differentiation in the context of this assay. So there are caveats to how one would interpret the readout from this, and exceptions to the rule. You can also ask the question as to whether patient cells are intrinsically able to differentiate into pharyngeal endoderm and thymic epithelial cell progenitors. For this sort of assay, you take patient fibroblasts, you make induced pluripotent stem cells, and you provide them with differentiation factors in vitro to force them to differentiate into definitive endoderm, anterior foregut endoderm, pharyngeal endoderm, and then thymic epithelial progenitor cells. And your readout here is whether they express transcriptionally the sort of genes you expect them to at each stage. And therefore, if the readout from this assay shows that patients' cells are impaired in their ability to differentiate in this way, one would be more suspicious of a thymic stromal cell defect and inclined to do a thymus transplantation. So briefly, just to talk a little bit about the defects that we've discussed today, Charge syndrome is caused by haploinsufficiency of CHD7, which is a chromatin remodeling enzyme. It's expressed in neural crest cells in the pharyngeal system during development, where it functionally interacts with TBX1 and retinoic acid signaling. This probably accounts for its phenotypic overlap with other causes of DeGeorge syndrome, and indeed patients can have that classic DeGeorge triad of heart disease, hyperparathyroidism, and thymic aplasia. They also have kind of lots of other different syndromic features, such as coloboma, defects in the iris or other parts of the eye, choanal atresia, and ear abnormalities. PAX1 is a transcription factor that regulates kind of cell fate specification in various organs and cell types, and including in the pharyngeal apparatus during development, and also plays a role later in thymic epithelial cell development. These defects in PAX1 in their relationship to thymic aplasia have only recently been described and have been shown to essentially lead to reduced transcriptional activity because they impair the way that PAX1 is able to bind to DNA. And when patient-derived iPSCs are tried, when you attempt to make patient-derived iPSCs differentiate in vitro into tech progenitors, this is impaired. It's part of a wider syndrome, autofacio cervical syndrome. So there are quite a lot of syndromic features, facial dysmorphism, ear abnormalities, and lots of different skeletal problems that also Graham mentioned. And importantly, these patients lack the DGS triad. So they don't have problems with the parathyroid or cardiac defects insofar as I know. And finally, just now to bring us back to some of the cases, I wanted to talk a little bit about FOXN1 defects. So George has talked about FOXN1 being the master regulator of thymic epithelial cell development and function. And as we all know, complete loss of function mutations in FOXN1 cause nude SCID. These patients require thymus transplantation for their survival. More recently, but increasingly, people are identifying hypomorphic FOXN1 mutations, often in the context of newborn screening. Various types of mutation are identified. The patients have a highly variable immunological and clinical phenotype, and we think this is likely dependent on the degree of residual FOXN1 transcriptional activity and the particular molecular mechanism at play. Importantly, these patients may lack alopecia and nail dystrophy, and as yet, their long-term outcome in management is unclear because they've only recently been identified. So now then to bring us back to the cases. So the second case that I presented with C-terminal mutation in FOXN1, we modeled their FOXN1 mutation. So we used for this a luciferase reporter assay in which luciferase expression is driven by the beta-5T promoter. This is a very well-known, well-characterized target of FOXN1. So when we transfect cells with the reporter construct and wild-type FOXN1, you can see that luciferase is expressed. But when we transfect the cells with the mutant delta 550 FOXN1, and we can see that this is a transcriptionally inactive protein as we predicted. But in order to recapitulate the kind of scenario in the patient, we actually also co-transfected the cells with equal amounts of wild type and delta 550 FOXN1, showing that actually delta 550 FOXN1 exerts a dominant negative effect on the wild type protein. Furthermore, if you now take a version of FOXN1 that we call delta 550b, so it stops at the same place as the patient's mutation, but has the canonical sequence between 457 and 550, so it doesn't have this frame-shifted scrambled sequence. You can see that again, this protein is transcriptionally inactive, but when co-transfected with wild type, it's not dominant negative. So the dominant negative effect really is coming from the scrambled non-canonical sequence. George mentioned the formation of these nuclear condensates in which we now know that FOXN1 operates. So we wanted to see how this mutant affected the formation of nuclear condensates. So on the left, you can see examples of experiments where we've tagged the wild-type FOXN1 with GFP, and on the right, where we've tagged delta 550 with GFP. So essentially, delta 550 FOXN1 is able to form nuclear condensates. When it's co-expressed with wild-type, as it would be in the patient, its ability to form nuclear condensates is not impaired by the wild-type. However, it itself impairs the ability of wild-type FOXN1 to form nuclear condensates when they're expressed together. And as George showed you with the photobleaching recovery experiments, actually the reason that we think this happens is that the delta 550 FOXN1 sits in nuclear condensates and does not allow that dynamic exchange for new FOXN1 molecules. And in this way, because it sits there for longer than the wild type, excludes wild type FOXN1 from accessing these functionally important nuclear condensates. What I didn't tell you is we actually did whole genome sequencing for this family and found that both the patient's older sister and father, who was in his 30s, both carried the same mutation. They were clinically thriving, although did have EBV viremia chronically, and they had a very similar immune phenotype with low T cells and lack of thymic output. So on the basis of having this family history, the Great Ormond Street team managed this patient supportively. And actually she's currently well. When I last heard that she was not on any immunoglobulin replacement or antimicrobial prophylaxis. The second case, the compound heterozygous patient with the FOXN1 variants, we modeled this allele again using the same luciferase reporter assay and confirmed our suspicion of it being a hypomorphic allele. And now I hand over to Alex to tell us about the management.
Thanks, Fatima. So yes, at this point, I think in particular, given the significant history of the patient who had a lymphoma and who after chemotherapy achieved like complete remission, we did reconsider. So initially we hadn't accepted this patient for thymus transplantation, but we then reconsidered because he achieved complete remission. He was clinically well, but remained with his immunophenotyping unchanged, so with absent naive T cells. And so we ultimately decided to proceed with thymus transplantation. So when he was 11 years old. And so that is now more than 2 years ago. And unfortunately, so he didn't achieve any significant level of thymopoiesis after thymus transplantation. At this stage, still has negligible naive T cells and very low TREX. So what you can see here from his biopsy at the site of transplantation, which was taken about 3 months after transplant, as we routinely do. So you can see that there is thymic tissue. However, it's relatively sparely, it's relatively empty. And what we do see are T cells that are CD3 positive, but are not CD1A positive, or if we take another marker that you saw in one of these other biopsies, it's not Ki67 positive, suggesting that they're not new T cells, and that basically there's no active thymic output at this stage. Then also the patient had another episode where he had a reactivation of his EBV, and again presented with some degree of lymphoproliferative disease and required another course of rituximab. So this is where we are at right now, and the patient is being managed by his team in Slovakia, but we remain closely involved in the care and in the discussions around how to further manage the patient in the future. And so I guess, as the previous case has shown, even when there is a thymic stromal cell defect, and even if you have some functional data showing that the defect can be significant, and similarly in this patient we have a PAX1 deficient patient, it's not always easy to make the decision in terms of treatment. And so for this fourth case, so in light of the high viral load for EBV, we had actually recommended to start with rituximab. Meanwhile, the local team had already started treatment with acyclovir and actually achieved some reduction in the EBV viral load. Plus, we're in the process of completing the workup of this patient. And when they performed a CT chest, they found some nodular consolidations in the right upper lobe. They then did a bronchoalveolar lavage and did some biopsies and found, probably an EBV-driven lymphoma. But these are very recent findings that need further confirmation. But at this stage, so it seems pretty clear that the patient is not ready for thymus transplantation and is not ready for BMT at this point either. And so again, I think afterwards, when we open up the discussion also to the panel, or in terms of the questions or suggestions that can come from the audience, it would be interesting to see what people think. But so to finish, in terms of maybe to summarize, basically the learning objectives we were aiming for in this grand round was, one, to appreciate the complexity of the thymic stromal cell compartment, to understand the different genetic etiologies of inborn errors of thymic stromal cell development. We have shown that that newborn screening for SCID will have an impact also in the diagnosis of novel thymic defects, and the importance of novel tools, in particular when we have genetically undefined defects or novel defects. And then to understand basically the indications for thymus transplantation and the expected outcomes after thymus transplantation, and also to appreciate the importance of early diagnosis and really referral for treatment.
Great, thank you very much, Alex and Fatima and George and Graham. So we're going to leave it, open this to the panel discussion now. And I also have great pleasure in inviting Aisling Flynn, who is an ACL in Newcastle, to the panel, to Isabelle André from Imagine in Paris, and Adam Klopke from Prague. So thank you for coming. Aisling's going to ask the questions, and if you've got them, can you please put them in the Q&A? But I'm going to kick off with Chairman's prerogative and ask a question for Isabelle and a question for Adam. So Isabelle, who should we refer for in vitro thymic testing, and where do we do that? How do we do that? And a question for Adam, which you can think about and you might want to discuss with the panel. We've heard about the Georg type transplants. Yeah. Should we be expanding the program to patients that maybe have failed BMT with GVHD of the thymus and no longer have thymic function? So Isabelle first.
Yes, thank you, Andy. So yes, we have performed several T-cell in vitro T-cell differentiation to diagnose patients who are T-, B+, NK+. And in fact, most of them were conclusive, so we could detect by this in vitro 2D differentiation systems that there was indeed an extrinsic thymic problem, so they required thymus transplant. But we had that case of T- B low patients in which for which T-cell differentiation was very good in vitro, and in fact she had started conditioning for haploidentical stem cell transplantation, and T-cell differentiation occurred. So she got naive T cells after a few months. So in fact, this patient was not appropriate for thymus transplantation, and it was an ADA case as described by Fatima. So in vitro T-cell differentiation is a good diagnostic test. It's quite fast. In the 2D system, we can get the answer within 3 to 4 weeks maximum. ATO required a little bit longer, but we are it's quite easy to do, and we have good results. So except for AGA, apparently it's a good test to diagnose these patients who require thymus transplantation.
Thank you very much, Isabelle. So Adam, sorry, I gave you a corker of a question there. Have you got any thoughts?
A little bit. Just to follow up on Isabelle a little bit, if I may. So this is a very cool assay. Is it necessary for those of us who maybe are thinking about assessing the T-cell differentiation potential in our patients at your lab? Is there any preparation for the patient necessary? What kind of material should people think about that they would need for you? So
for the 2D differentiation system, we need bone marrow CD34 positive cells. Mm-hmm. So we don't need a lot of bone marrow sample. 1 ml is enough. We also worked with 500 microliter of bone marrow sample. Then we sort the CD34 and we do the test. We just require 20,000 CD34 positive cells to do the test. Mm-hmm. And as control, we usually use cord blood CD34 and mobilized peripheral blood CD34.
Sure. Okay.
So just the bone marrow sample.
Yeah. And to your tricky question about expanding the indication criteria for thymic transplantation, I suppose. I mean, I'm not the one who does these, so that's more of a question to Graham and Alex, I guess. But I mean, depends on what the underlying genetic cause is, doesn't it? I mean, if it was a previously bone marrow transplanted patient who has a primarily thymic defect, then once the bone marrow stops being able to produce or proliferate the mature cells which have nowhere to mature, then for sure I think the transplantation of the playground for the developing thymocytes is definitely meaningful. If it's for other indications, then I would defer to the experts at GOSH, to be honest.
Okay, thanks. Alex, Graham.
Well, on that point, I mean, I think I hope it came out of what I presented that actually these patients are prone to immune dysregulation problems and inflammation. And I think starting a thymus transplant on the patient to rescue them from graft versus host disease is nice theoretically, but in practice, I think it's probably going to be as very likely to be complicated by worse immune dysregulation. If the patient has got GVH, there must be some mismatch somewhere, even if you can't find it with the standard antigens, between the HSCT donor and the recipient. And as I mentioned, if you then have a thymus putting in another lot of HLA types that doesn't match those mismatching antigens, then you could just end up promoting the graft versus host disease. So I think if there was to be an extension to indications, it probably wouldn't be for failed transplants with severe chronic graft versus host disease of the thymus. I think there may be a case in occasional patients who've had total thymectomies early on in life and actually are running. There are data that suggesting that they do get increased immunopathogenic pathological problems. Replacing their thymuses might be another thing to think about developing.
Graham, I know that this is not directly related to PID, but what's your opinion about the co-transplantation of thymus with parenchymatous organs like lungs or heart to facilitate the tolerance, better tolerance of the transplanted organ?
Yeah, I think theoretically that offers, if you can get organ donation from of the thymus and an organ from the same donor, theoretically that will work. Practically, that's quite difficult. Where harvesting thymus tissue from young infants who've got big juicy thymuses, the sort of organ donors that one's likely to get are going to have atrophying thymuses because they're older, possibly because they've been sick and they've been dying of something, which is why they're donors. So yeah, I mean, it's an attractive thing for the future, but there's quite a few practical hurdles to overcome, I think.
Thank you.
Maybe, Graham, just in completion of this, and this wasn't mentioned, but in terms of the patients with hypoparathyroidism, so as a proof of concept also for the question which was just asked, so we've had, for example, one case where there was co-transplantation of thymus tissue and of parathyroid glands that were ectopic, that were in the thymus. And in this one patient, we, as a proof of concept for tolerance developing towards the solid organ, you can see that this patient was not only cured of her immunodeficiency, but also of her hypoparathyroidism.
Okay, so shall we start to have a look at some of the questions in the Q&A? Starting from the top. So how low T cell levels can be tolerated in partial to Georg? And do you think that the indications for thymic transplant should be expanded? So Graham or Alex, would you like to touch on that?
Do you want to go, Alex? I'm very happy to talk. Well, I think it depends. What do you mean by T cells? I mean, quite a few of our complete de Georg patients have quite a number of T cells sometimes, which are oligoclonal and have no evidence of having come out of a thymus. So in that situation, apparent partial DiGeorges might be close to complete DiGeorges. If a true partial DiGeorge patient will have a little bit of thymus, a hypertrophied thymus rather than athymia, and that the issue then becomes yes, they may get autoimmunity, they may get infections, but if to think about a thymus transplant, you've got two issues. One is the bit of thymus they have may compete with the precursor cells for where they go. And the second thing is that there may be sufficient immunity to reject the thymus. I didn't mention in my talk, but we've had one recent patient who seemed to be a complete DiGeorge with no T cells at all, but nevertheless managed to reject the thymus, and we showed double-stranded, we showed donor-specific antibodies against the donor HLA antigen. So a number of things to overcome there. But I think the important thing with these severe partials is to really assess them to see whether they really are, they're masquerading as partial because they've got a few T cells, but actually they're nearer complete. And that would be with looking carefully at RTs and naive cells and TRECs and so on.
I think in completion then of this, if we look at the results that we have right now, also for in our patients, and Graham has mentioned this, where these complete DiGeorge patients or become partial DiGeorge patients. So certainly for partial DiGeorge patients who are not like too severe partial DiGeorge patients, there would be no point really at the moment to proceed with thymus transplantation, because we wouldn't be able to really say that we'll be able to improve their immunology with the current modality of treatment.
Okay, great. So the next one, so a lot of the questions are sort of transplant related, but the next one is about, have you studied if host mesenchymal stem cells start to differentiate it into thymic epithelial cells in the transplant? And if so, it could explain why HLA matching is not always vital. And another question that's sort of a continuum from that is, are host dendritic cells infiltrating the transplant? And is there any correlation to transplant success and how many dendritic cells are there, or how large the epithelial compartment is? So again, maybe Graham, Alex, maybe best to answer this question in terms of what we know about the histology of the transplants after they're done.
I'll kick off again. I think those are very interesting, fascinating questions, and we would the simple answer is no. We haven't studied mesenchymal stem cells in the transplanted thymus. The biopsies that we get are terribly small, and they are buried. What the surgeons do for the biopsies, they take a piece of muscle where they put the thymus, and so most of the biopsy that's taken out is actually muscle, and then we've got to try and find a bit of thymus in it, and sometimes they miss, and so there isn't any thymus there at all. And it does make studying them extensively quite difficult. Yes, one can do normal histological sections and cut through the thymus and stain with things, but doing more complicated things is quite difficult. And yes, we'd love to know about, the role of the dendritic cells migrating in, and that's, of course, one of the theories that we have for why a mismatched thymus is able to, in general, produce self-tolerance, although there are breakthroughs, as I mentioned, in terms of thyroid disease and cytopenias. In general, there is post-tolerance, and so those dendritic cells, post-derived dendritic cells, may be important in the antigen presentation for negative selection. Alex, anything to add?
No, just yes, it's a fascinating question, and we're very interested in looking into this further using some of these novel research techniques, including some that Georg has shown in his presentation, which were applied to normal thymus, but which ideally we're hoping in the future to be able to apply to these thymic biopsies to then learn more about the composition of these biopsies.
Okay.
If I may, this might be a nice follow-up for Georg. I'm wondering what is the most up-to-date information on what the role of the DCs migrating in as compared to the MTEX and CTEX is. Do they present a different set of antigens for the developing T cells? How are they special compared to the TEX?
Very good question indeed. We know that the composite of the cells able to negatively select thymocytes includes not only thymic epithelial cells in cortex and medulla, but also hematopoietic cells, in particular B cells, dendritic cells, probably some macrophages, but also mesenchymal cells, at least in an experimental setup. So what has been shown, and again experimentally, because the thymus is just not a great organ to do clinical research while the patient is largely unaffected, what has been shown experimentally is that B cells, particularly B cells from the germinal center, can actually re-enter the thymus, as do T cells. And they could be shuttling peripherally taken up antigens back to the thymus for presentation. Under experimental conditions, you can also show that certain antigens are much better presented for negative selection by dendritic cells than by thymic epithelial cells, whereby dendritic cells take up from dying or from secreting thymic epithelial cells the antigen, process it, and then present it in the context of their own MHC. So this is clearly an interplay between the epithelial and the non-epithelial stroma that collectively then shapes the T cell antigen receptor repertoire.
Are there any mechanisms which would prevent a peripherally obtained maybe alloantigen to get into the thymus and then create tolerance towards it, be it pathogen derived or a foreign object derived or anything like that? Because I can imagine in this setting that a DC or a B cell that has encountered something that is not own to the body would then go into the thymus and start tolerating.
Yes, so that is thought that this happens obviously after bone marrow transplantation, where the new haplotype of the donor bone marrow then is represented in the hematopoietic compartment of the thymus and provides part of the successful tolerance to donor tissue. We know from tetraparental experiments that actually most likely there's a hierarchy of keeping tolerance in place. And the thymic epithelial compartment is not as efficient in maintaining deletional tolerance as probably most of us wish to assume. And so there is an escape of autoreactive T cells in, again, experimental conditions that you can easily show, whereby then these autoreactive T cells are tuned in the periphery not to be responsive. So you shouldn't think of the thymus as the everything and do everything, although after 35 years in that field of research, I believe they do quite a bit, but there is a periphery that fine-tunes some of the inadequacies of the thymic microenvironment in shaping the T-cell antigen receptor repertoire.
Thank you.
Okay.
And with regards probably to that question about mesenchyme and can they turn into thymic epithelial cells, the beauty and also trickery of single-cell transcriptomics is that you can actually interrogate the relationship of different cell lineages at different stages of their maturation based on the transcriptomic complexity that individual cells show. And then you can basically line up these cells where you only look at small differences between a stage 1 and the subsequent stage. And if you do that, there is no indication that epithelial cells of endodermal origin actually become mesenchymal cells in normal development or the other way around. Now, there are some thoughts and some experiments have made a strong case for that there is epithelial mesenchymal transition with age, but that is a different kettle of fish.
Okay, and the next question is, for patients with complete DeGeorge syndrome and thymic transplant, so do you see more autoimmunity issues compared to patients who have hematopoietic stem cell transplant? So I don't know if that's been directly compared. I can't imagine numbers are huge to compare, but Graham or Andy or Alex, would you have any knowledge on any comparison?
Well, the simple answer to that is yes. We do see more autoimmunity. As I mentioned, I think during the early reconstitution phase, there's maybe transient dysregulation and it will subside, and that is seen in HSCT as well. But after HSCT, you will hopefully end up with a pretty normal immune system, whereas with ours, we still end up with a partial DeGeorge picture where possibly the susceptibility to autoimmunity continues. The question has to be asked is whether a lot of our autoimmunity is directed against the thyroid, and there are a number of publications going back over the years showing that partial DeGeorge patients have a high incidence of thyroid autoimmunity. So one question is whether the Thymus transplant's got nothing to do with the development of autoimmunity. It is just these patients are DeGeorges and they were going to get autoimmunity anyway. Yeah, so the answer is yes. Don't know if anyone, Andy, you've got anything to add to that?
No.
Okay, so for patients who do not achieve good T-cell reconstitution after their thymic transplant, what is the next step? So is there any role for stem cell transplant or anything else?
First of all, some of these patients with very low level of immune reconstitution, actually after thymus transplantation, they do remarkably well clinically. So for these patients, certainly we've just taken the approach to monitor them, but you know they do remarkably well. Most of, I don't know, Graham, if you want to add to that.
Yeah, so some patients have got pretty low numbers, but are clinically okay. I think we've repeated 2 transplants because they didn't work. One was the one that was rejected, and the other was lost early on due to septic shock. And they both worked the second time. So it is possible to repeat the thymus transplant if you think that things might go better the second time. And then we haven't touched on this, but in a dire situation, say with an uncontrollable systemic viral infection such as an adenovirus or CMV, if there was a matched sibling donor, then you could then there might be a case for a rescue hematopoietic stem cell transplant, which will give some passively acquired T cell immunity against whichever virus.
Thanks, Graham. I think in the interest of time, we probably ought to wrap this up because I think we've gone slightly over time. Thank you to the panelists and to the speakers. I think it's been a really interesting session. And hopefully we can have another thymic session in the not too distant future. Thank you and good evening.
Bye.
Bye.
Thank you.
Bye-bye.
Thanks.